to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 39th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Moreno, and I have been in the LBA and cell-based assay space for over 26 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I have been in the bioanalytical LCMS space for over 26 years.
John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas. John is in upstate New York, and our producers are in Missouri and Kansas City. So we are all over the map. We are excited to have you listening to the 39th episode of the podcast, which is now available virtually everywhere. Apple Podcast, iTunes, Spotify, Google Podcast, Amazon, and many others. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. As many of you know, we started this podcast because of COVID restrictions preventing us from continuing business as usual. We were unable to meet face-to-face, but we wanted to continue to communicate with all of you. The podcast was an idea we'd always had, but it suddenly became the perfect medium for us to employ. Today's podcast, we have a very special guest. We have our business development representative from the Mid-Atlantic. Her name is Mallory Jolly. Please say hello, Mallory.
Hello, Dominic and John. Thanks so much. And, I'm really thrilled to be a part of this episode with you guys. Looking forward to it.
As always, the podcast will be a review of the latest news and resources. Typically, we talk about COVID and then non-COVID, and then we got some big investment news today. Very big investment news. Big show. Not only do we have a special guest, but we have some amazing investment news that we get to discuss today. But then after the news and resources, we'll focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we will wrap up and give you a little teaser for the next episode, letting you know what is coming in the future. We're looking forward to a fun episode today. To kick us off for podcast number 39, John is going to go over today's podcast topic a bit before we jump into the news and resources. John?
So, as usual, we'll kick off with the news, going through COVID, non-COVID news, and our big investment announcement. Our main topic today is actually, we're calling it an Introduction to Bioanalysis, Questions from a Beginner. This is really a discussion about how do we as people who are routinely doing bioanalytical work, how do we communicate what we do to people who are unfamiliar with what bioanalysis is?
Great, John. You want to kick us off with the news?
Sure. This first item is actually probably on everyone's lips. We weren't actually originally going to talk to it, but it popped up very, very quickly. Is that the Fed call halt to J&J COVID-19 vaccine rollout after extremely rare blood clot cases. In this country, they've administered 7 million doses of the J&J vaccine, but there's been 6 people with this rare blood clot has occurred, and it's all been 6 white women, all between 18 and 48. One actually died, and one's still in critical condition. So the FDA and CDC have put a halt on it while they investigate this. And a similar thing had happened in Europe, where EUA had done a similar thing where they'd observed this with the J&J vaccine while they're digging into it. And like I say, I have to emphasize this is very rare. I mean, 6 out of 7 million is about 0.001%, but it's obviously trying to determine what the risks are with this vaccine.
Yeah, John, I think if this was such a hot topic, I don't even know, standard sort of vaccine for, I don't know, Zostra or some sort of other sort of, HPV vaccine. I don't even know if it'd be making headlines like this. I think part of it is due to it being so much of a focus on COVID. And just personal side, I was scheduled to get the J&J vaccine, John, and then I did cancel it and subsequently went on and got a Pfizer shot recently, which I'm very excited about. But I do think that this is, from a, like a world health, we really hope this vaccine pulls through and that this is just a temporary halt. From what I can tell, it didn't seem like this was going to kill the vaccine in any way, shape, or form. I do think, I think you'll see it come back. I think it's due to the nature of it. Again, it's very stable, right? There's just a one vaccine shot. It doesn't
Sure.
require that cold chain and all that. But maybe the other rounding out piece here is, I think the manufacturer is at fault, but maybe I should be careful there. Let me restate
No,
what I just said.
no,
I
I
think
would.
the manufacturing,
No, I mean have been manufacturing issues that we do want to talk about in a
yeah,
later podcast. Um,
yeah.
but
Maybe
I
we'll
think,
press pause on that. That's a good point, John. But I
yeah,
think it's not. I want everybody in the audience to understand this is very little to do with the actual vaccine and more to do with maybe potentially some human error, right, John?
I
And
don't
from
honestly
a
we can't say. I mean, we honestly don't know. I mean, the FDA thought the halt would be temporary, and they've actually extended it to next week. It's interesting because you know the actual this condition is coming like 1 to 3 weeks after vaccination, and all of us who've been through the vaccination have had symptoms a day or two after, and then we felt nothing. So trying to tie something that happens 1 to 3 weeks after administration of a vaccine in such a small pocket of the population to say what the factor is here is a difficult. I mean,
Yeah,
it's a tough thing to do. So, I mean,
yeah.
we
There's
just
that.
don't know what it's
I
tied
agree.
to. And actually, I read somewhere on social media that instance of blood clots in actual COVID patients is a significantly higher percentage than 0.001%. So the vaccine
Yeah,
is still beneficial in terms of that. It's just, I think it's an overabundance of caution, which is fine. I've got no problem with that. It's also we're in an age where we have so much information, and the COVID is so dominant. There's so much focus on it that something like I agree. No, a standard vaccine might have this happening, but no one will pay any attention to it. But because it's a COVID vaccine, we're all riveted to it and like what's
yeah,
going on. And yeah, we seen that. We saw that when the AstraZeneca trial halted. We've seen it, in the early days when there was possible cases of anaphylactic shock to the Pfizer vaccine. It's just such a heightened awareness,
yeah.
which we will talk on later about these vaccines and what it means for us going forward.
Yeah, I mean, I think the other piece, and we'll maybe I think future podcasts will address it a little more. It's just the whole manufacturing issue that
Sure.
emerged
Yep.
prior to this gives that
Yep.
just implies it. So my mind goes immediately in a different direction. And then even this type of vaccine is it's grown in a baculovirus. It is a virus-like particle. A lot of passion there, John. I studied those a lot. And so when you hear of these types of responses, it could be just due to some impurities, as well as maybe some sort of agglutination of the vaccine itself, which again, in my mind, is more some sort of human error. But again, that we don't know that. I'm not saying that, but there is some indirect evidence there that gives us all a little bit of pause. But
Yes.
very again. I think we're both in the court of when this gets off of hold, or for those of you that have had the J&J vaccine, I wouldn't worry about it. I think you're very protected. I think it's a very good vaccine.
Mm-hmm.
Me
Yep.
personally, I got the J&J vaccine about, I think it was 3.5 weeks ago. So I'm in the green zone here. I definitely had some symptoms, that night. I think my fever was around 101, but by the time I woke up in the morning, I felt great, and I've been fine ever since. So, I'd be interested to see. I did read with that specific blood clot that it was causing. Giving heparin, which is the normal treatment for blood clots, it actually made it worse, which
Yes.
is really
Yep.
Yeah.
interesting. So,
Look at you, Mallory. Very well done. And like I said, you're safe. But no, you brought up a good point about the heparin. So there's a lot of TBDs in there, to be
Yep.
determined.
yeah.
Yep. Yep. So let's move on to the next topic. This is so the NBA have dealt actually with COVID very well. They suspended play in March last year. Then they pretty much set up a bubble for the teams that were closest to qualifying for the playoffs in Disney World, and went through playoffs, etc. Finished their season with very few infections or cases of COVID. So really did a good job of protecting their teams. Well, now the next step for the NBA is they are looking to install Clear Health Pass Tech as arenas refill. So it's how can they get crowds back but still maintain protection. And Clear Health is effectively it's an app on the phone that says what your history has been in terms of vaccination and things like that. So as a fan, you can present that and be allowed into arenas. So it's interesting how much the NBA is thinking about this as they move forward.
Yeah, it's great, John. I think this is me personally. I got nothing to hide, so this would be very good to me. I like this idea. It's going to make me want to go to an NBA arena. Looks like it's also this technology is kind of being used by the state of Hawaii, MGM Resorts, the 9/11 Memorial, and the Founders Table Restaurant Group. So there's a few others I think kind of
Yeah, I
piggybacking onto this, but clearly the NBA dominates the scene here, and I think that I hope we see some sort of, if it's not just the clear technology, something that allows us to go. I would love to go see a Sporting KC game or some sort of football game or something like that that we can just feel a little bit more comfortable. But this is great stuff. The NBA has done a very good job, John. You're very right.
And I'm sure this is another topic for another day, but how sustainable is it to just have these paper cards where, like for the Hawaii one you mentioned, I saw that you just take a picture of it and then upload it to an app, and then that's how, you're cleared to go. How sustainable is just that paper card? I mean,
Well, it's not. I think so. I saw somewhere about people laminating them, but when I had my second dose, the person who administered it basically said, "Don't keep your card on you because my wallet is small, so it was already distorting the card. So take a photo of it and have it on your phone." So that's what I've done. So the card actually never goes anywhere with me, but I do have that record of being vaccinated if anyone asks.
Yeah, but I don't know. This is a topic that's a little bit I've, as I said earlier, I just feel like we all should be having some sort of vaccine passport. And
mhm.
if I'm a restaurant owner and I want you to have a vaccine, you got to show me your passport, whether it's on your phone or whatever. Then, but I think there's making it a mandate and all this sort of stuff. We're not going to talk so much about that. But to Mallory's point, it's sort of to be determined. But I do think you should be kind of prepared to like have a license, right? We got a license, and you could see
I,
maybe somehow having something on that.
And on a similar topic, I mean, when we travelled, to Madagascar again, when we went there 2 years ago, my son and I, we actually got a vaccine passport. It's like a little booklet that details all the vaccines that you had administered before you went into the country. No one asked for it, but we had it in case they did, because there was a record of vaccines that they wanted you to have before you could enter the country. So I mean, it's something that travellers routinely have done, to have a passport handy.
Yeah,
And
I
actually,
don't know what
another interesting note. When we were away 2 weeks ago, we went went down the main street of the town we were staying in, and there was one shop we went into, and the owner had basically put. He'd photocopied his vaccination record and mounted and put it on the front glass door of the shop, to say that he'd been vaccinated, and he wasn't wearing a mask in the shop while all the customers were. So I just thought it was an interesting approach to that one.
Yeah, no, and now you're getting into something that we're not going to talk about. But if you're vaccinated, do you have to wear a mask? But no, it's good. It's really the NBA story and this whole thing. It's great to be talking about this.
Sure.
Roughly a year into COVID. I mean, well, how great it's been scientifically, and just keep wearing your mask though, and keep trying to social distance even until we get a little bit more information. Right? There's a lot of swirling stuff out there around.
Yep.
Are you protected or not? So, but John, maybe we'll move on. I think the next story is a great one. I love a good chicken egg story, John. Tell
Well,
us a little bit about
yeah. So it's basically researchers are hatching a low cost COVID-19 vaccine, and this is like really, to me, this is the really crucial next step. It's basically researchers looking of a way to get low cost vaccines so we can actually get it across the globe, because we've talked numerous times about the imbalance in terms of vaccination rollout. We're all happily talking about how we're either fully vaccinated or partially vaccinated, but there's countries across the globe where no one's vaccinated because the Western countries cornered the market on available doses. Oh, they're expensive. So something like this is great, and it's a new vaccine, NDV HXP5. Is entering clinical trials in Brazil, Mexico, Thailand, and Vietnam. And so do you want to go on the chicken egg piece, and we can talk a bit more about the background, because it's a great
Yeah, I
story.
mean, chicken eggs are used for I think the majority of the influenza
Yep,
vaccine.
exactly.
I feel like there's been some advancements there, but they still use chick eggs. And then I think even maybe Mallory could relate to this. We make a lot of antibodies in chicken eggs these days because they make great positive controls in ADA assays. See, I turned on you, John, a little bit, but that's why we love a good chicken egg story, because they make these immunoglobulin Ys. And this is a big thing for John Pirro, our senior director, one of our senior directors. He loves this concept because, if you're trying to get a anti-drug antibody, you're trying to detect a human immunoglobulin. These chicken eggs are so vastly different from, say, rabbits and other species. They're a little smaller, and they make for great positive controls in drug development for ADA assays. But John, maybe we'll get back on topic with the need for chicken eggs. And obviously the utility of it is the cost, right? It's a
Yeah,
really,
absolutely.
any country, I got a buddy who had worked in Nepal, and he raised a bunch of, actually just recently, they made a bunch of vaccines and eggs there as well for helping with Newcastle virus, which just really can hurt chickens.
Which
And so
actually
we're
plays
talking
into the
about them having to
story.
stay alive, right? If your chickens are dying, you might die. So they were able to produce a massive amount of vaccines in these chicken eggs. So great story, John. But yeah, share more thoughts. Yeah.
Yeah, this is all. It's all about vaccine optimization, and really it shows how critical it is to learn from current virus for future viruses, because a lot of the work that is actually occurring here is building on some work that was done by a guy called Jason McClellan, who's a structural biologist. At the time, was at Geisel School of Medicine at Dartmouth, and his colleagues, and they were actually working on MERS, and they targeted the spike protein in MERS, and found what researchers have talked about with the spike proteins. It changes shape. It goes between the tulip shape and the javelin shape. If it's in the javelin shape, then antibodies and things are ineffective. If they can make it stay in the tulip shape, then they've got a way of attacking it. They found that if they did a couple of substitutions in the spike protein, they could keep it in the tulip shape. And it's actually just 2 amino acids. They put 2 prolines in, and that was their first step on this. Once you got to COVID, the research didn't really go anywhere because MERS didn't go anywhere. But now with COVID, it actually turns out that all the vaccines that are authorized so far have all taken advantage of this 2 proline switch to make them effective. Meantime, this guy McClellan has gone back. He's thought if 2 prolines made a difference, what else could make a difference? So done various switches, and they've actually now at the state, they found that the optimal was actually 6 different proline switches within the protein, and they called this hexapro, which is now the basis for this additional vaccine, and that's where other people have got involved. And it's funny you talk about the Newcastle disease, because it's actually building on work that was done by developing Ebola vaccine. By adding the Ebola gene to Newcastle disease virus's set of genes and growing it in the egg that way, and that's how the chicken eggs come into this.
Yeah. No, John, what a clever group. Fascinating. I don't know how much. I hope the audience understood and appreciated the way they found the tulip-like and then the javelin-like. I mean, conceptually, you don't have to be a biochemist or a chemist to understand one's shaped like a tulip, one's more linear, right? You stick these prolines in, it keeps it tulip-like, and then they build upon that. They go from 2 to 6. I mean, this is just great science, John. I mean,
Yeah,
this is the
absolutely.
stuff that
It's a great article, and
yeah,
yeah. So like I said, so this vaccine has been shown to be very effective in mice and hamsters. It's now trialing in like 4 countries that I think I mentioned,
yeah, and it's
Thailand,
great. I mean,
Vietnam, and Brazil. And
it's
so it's phase 3 will come later. And it's the big deal is the nice thing about this is it's cheap and it's localized, so it
yeah.
actually gives countries the ability to develop and administer vaccines that are produced in their own country. So it could be a real game changer when it comes
Yeah,
to actually
absolutely, John. And they even
globally vaccinating.
the article even alludes to how a lot of countries, I call them resource poor, I guess, they
Yep,
actually have some of these hatcheries available to do this because of influenza, as we kind of alluded to.
yep,
So they have the
exactly.
backbone to make this. They just need the technology, and seems like it's on its way. And really, just a great, the egg actually does all the work, right? So it's
Yep.
really, really, I love it. Absolutely love it, John.
I will
It's
be
great
we'll be
stuff.
watching this one with extreme interest because, like I said, it's great to see that potential
Yeah.
for
John, we're
global.
going to move on to our next item, which is, TikTok famous. Not my famous, not my favorite website. I have done a couple TikToks with my 8-year-old nephew. So I
How
think
can I get my hands on those?
I don't know if you can. I think we had 42 views or something like that. It's the most he had, but it's a pretty funny one. But I won't elaborate too much. But John, tell us a little bit more about how some of our big, the Pfizers and the Modernas are
I,
getting
I,
out there, getting their message out there, and getting it to the youth, I suppose,
I
right?
think well, yeah, the headline was TikTok famous Pfizer, Moderna, J&J, and AstraZeneca shout-outs on social media boost vaccine credibility and pharma's image. And I think that I mean, if you're anywhere on social media, it's not just TikTok. There is a lot of discussion of the vaccines, and I think that what this talked about was the one particular TikTok video where it's a comedian was basically I think having a sort of face off between conversation between someone between the Moderna vaccine and the Pfizer vaccine, and I've seen similar things on TikTok. Sorry, on Twitter, where people will talk about I've got the Pfizer one, and people that follow each other. There's a sort of friendly rivalry building up because one's had one vaccine or the other, and I just thought it was interesting to touch on the way, and it all plays into that first news story about how much focus there is, how much our conversation has revolved around the vaccines when you're out in public.
Yeah.
I was picking up a food order last Friday, and this is a restaurant we've gone to regularly. It's just up the road in the village, and I just sort of said, well, a couple more weeks, well, everyone in my family will be fully vaccinated. We're going to come and start using the restaurant rather than just relying on takeout. And so the person who served me talked that they just had their vaccine and they got the J&J. I was on the Pfizer, and she was sort of talking about she went for J&J because of didn't like needles. And it's funny how it's sort of it builds conversations, and it's the way it has entered daily life where people wouldn't talk about this stuff before, but it's and like say pharma is now benefited from that on the basis of people know they're being helped, so it really does boost the image and reputation. And this, we talked. They have that 6 monthly update about the reputation that various bodies have in the public's eye. And last time we spoke, pharma had jumped massively, and this is all obviously feeding
Yeah.
into that.
And
Yeah, I think it's interesting. Oh, go ahead, Mallory.
I don't know if you guys have seen there's complete like social media groups dedicated to finding the vaccines. So they're called like vaccine hunter groups. And you can see them for the state, you can see them for the city, and it's
Oh
just a
yeah.
bunch of people like live time saying this location just opened up appointments for this shot. And that's actually how I got my J&J shot.
Same with me. Yeah.
Was
There's a Twitter feed you can get on, right? So I don't know if it's
Yeah,
similar to
yeah,
what.
mine,
Yeah.
I saw the Twitter one, there's one on Reddit, there's one on Facebook. I mean, it's everywhere. There's social media communities just built around trying to get these vaccines, which is
No, it's good.
that's really interesting.
No, I think it's fascinating that even the article alludes to how someone's 5-year-old daughter talking to
Yes.
her grandparents, asked about, did you get the Moderna? I mean, this is, like John said, this is becoming a thing where, regardless of age, the exposure. Think about a 5-year-old asking if you got a Moderna vaccine. That's unbelievable, right? That's awesome. Like,
Yeah,
there's so
absolutely.
much that makes me think, oh wow, people are, maybe we're going to get over some of these phobias and fears around vaccines if 5-year-olds are talking about it. But it also, I think, we're kind of coming out of COVID a little bit, and I think it's a good way to kind of, everybody seems to be rallying around it. Although I think there are still good section of the population that may never take the vaccine, but it's
Sure.
it's really a fun story. It's a fun
Yep,
story.
yep.
Mhm.
So I'll move on to the last of the COVID stories, which is actually another one that's near and dear
Yeah.
to my heart. And actually, it's sort of a follow up to a question that was asked by Chris Frankovich's daughter in a podcast several weeks ago, where she was talking about vaccination of animals, and the headline on this is variation in predicted COVID-19 risk among lemurs and lorises. And basically the story here is we know that SARS-CoV-2 basically attaches to the ACE2 enzyme, so angiotensin converting enzyme 2. So this group of researchers actually looked at the ACE levels in a number of they're quite primitive primates, the lemurs and lorises, and they looked at 71 species of them to look at levels of ACE2 and use that as a predictor of possible vulnerability. And so of the species that they looked at, the groups that they were most concerned about are the II, which is very threatened. They're all rare and threatened lemurs, woolly lemurs, and then sharks. And it plays into, we talk about the cheap vaccine story just a few minutes ago. It plays into that. They're for somewhere like Madagascar, where these animals are native, they don't live anywhere else. But there's a large portion of the population of the island are dependent on tourism because of these animals. So if COVID-2 translates to the animals, then it has an impact on the economy, which is already a very poor economy. So it's like, this it's a much more far-reaching issue than just an animal might get sick. It's what would be the effect if they did, and then how can we prevent against it? So should we be? So the author's take was, as a conservation priority, we should be vaccinating these species as soon as possible. It's a very interesting, conundrum.
Yeah, I hope we do that by feed, right? Are you going to line up the lemur and try to inject them? Maybe we are, but I think you could maybe make it in a food pellet or some sort of feed. But no, it is, John. I think it's great that Chris's daughter had asked us, well, are animals susceptible? And you found this, so I hope she's listening and understands that it looks like people do care about the animals and they're researching it. But of course, John, I'm going to move over to the ACE2 receptor, right?
Sure.
That's where I think, you know. That angiotensin converting enzyme 2, I wonder how well that's been studied, and I haven't seen much recently just in humans, but I do think it can play a role in, like, can you get COVID? Some people are asymptomatic. Why are some people getting a vaccine and then the second one and getting terribly ill and others aren't? But no, I don't know. I think that might have some to be determined. And even yesterday, while I was working out, I had this. I know sometimes I was swimming, John, and so when I get in that pool, sometimes my mind drifts, and I was literally
Yep.
just
Yep.
thinking about ACE2 and how I wonder if, very much again, I revert back to HIV and the CCX7, I think, is the receptor that was mutated that showed prevented, infection, and I wonder if it's not there, maybe we'll evolve too. So I think there's some, again, my mind really drifts out there. I wonder if over time, 5 years from now, maybe there's some um, changes that can occur to help you, potentially prevent getting this type of,
Sure.
as
Yeah.
there's more variants and all of that, you wonder if there's some evolution in there. But maybe I'm just
So, no,
in a
but
good mood this morning,
it,
John. Maybe I'm getting too philosophical today, right? Maybe I'm
no,
too
it,
Darwinistic today.
sure, but it is interesting though, because if this group looked at levels of ACE2 as an attempt to predict for lower primates, but could we do that for people? If you're measuring ACE2 levels, well, is that an indicator of
Yeah.
susceptibility? Well, we
Yeah.
don't know. This article doesn't know that. They're just saying this is our potential concern based on
Well,
what we're seeing for these animals. And so, obviously, I don't want to test it and see can we administer it to the animals to back up their theory. But
yeah.
it's question longer term: is that something that could be a useful test for for people to say, yeah,
Yeah.
you haven't got it, but you've got high ACE2 levels. You want to be a little bit more careful.
Well, I think you touched on really another very important thing, the level of them, right? The density.
Yeah,
Is it good
yeah,
to have, well, maybe it's bad to have a lot or good. We don't know, right? But
yeah.
that's going to be, and that's a little bit tougher to study, right? That might even take some flow cytometric analysis to look at how many are on a given cell. But I hope to see more about that. But John, because I think we're running a little long.
Sure.
Let's go over because we got some real cool investment news, don't we? But let's wrap up a couple more the non-COVID news, John. I think we got a couple. We're running a little short of time. Maybe just pick one of the two. So
And that
which
if
one
it's
you want to
on,
go with?
well, actually, let's go with the brief one in that case. We'll save the IL-2 for next time because it
Yeah.
is a long involved story, but again, it was
Don't
a
get me on IL-2, John. I've been studying that
so
for 25 years. You
so
can't do that in 30 seconds. So let's move on to the other one.
so we'll do it next time. So this one I thought was if anyone who suffers from seasonal allergies, and at the moment I've just started taking Montelukast because it's seasonal allergy season for me, and it only lasts a couple of months. And so even though it's snowing today, I still I'm taking my daily dose to make sure I'm not struggling with itchy eyes, etc. But this doesn't actually affect the US yet. But J&J actually have an allergy fighting contact lens, which has just been approved in Japan. And so, and if you wear contact lenses and suffer from allergies, it can be utterly miserable. If you put your lenses in first thing in the morning, you're waiting for your allergy medicine to take effect. That itchy eye thing can be awful. So they actually have J&J developed AccuView TheraVision lenses with Ketoifin, that are basically daily disposable contacts with an anti-allergy medicine in the lens to help move forward. I just thought it was like a really nice little piece of technology.
It's so cool. So,
Yeah,
I just recently got glasses maybe 2 years ago, and I don't have contacts yet because I can kind of see. I really got them because I didn't want to squint anymore and give me wrinkles and make my head hurt late at night. But no, if they came out with a contact lens, because I am starting to get some late age related allergies. I never had them. As I get older, I am getting more of it. This would be something. If this came out, I might convert to contact lenses. I love this type of stuff. It's great
yeah,
technology. It's like sci-fi movie technology. Put something in your eye and it cures you. Right? I
sure.
love it.
Yeah, yeah.
Absolutely love it. It's a great story, and hats off to J&J. Really a leader in the eye space. They're a
I,
very
I
big
have
leader
to.
in this space.
I held off on contact lenses for years, and then once I put them in, there's no way I'd
Yeah,
ever go back to glasses
I'm
again.
still dabbling with surgery, right? Like
It's
it's
like,
just
yeah.
not bad enough. Yeah.
So
But
it
John,
was,
no,
yeah.
that's John. We're both kind of anxious to talk about this next topic. There's been some very big news in the investment world. I mean, this is huge news for us, John. Right?
Yeah,
I'm going to let you do. I don't know if you want me to do it. You can do it. But we've got some incredibly. If you haven't heard already,
yeah.
if you have not heard, we're going to sound the alarm. We're going to ring the bell. John, what is the big investment news?
So this is now 39 episodes of this podcast, and we've done news about all these other companies, and we've never mentioned KCS in the news. So yesterday we sent out a press release to say that we now have a new majority investor, Vitruvian Partners, who are basically a and the growth focused investment firm have acquired a majority ownership stake in KCS, and we're all really excited about what this might mean for the long term development of the company, because we're all committed to KCS and the growth vision of the company. But this will really help us move forward into the future.
Yeah, so a couple things, right? Vitruvian is the company who bought a majority share of KCS. But what's really cool about all of this is we had three existing ownership groups, and all three of them are reinvesting back in the company, John. So it's not like they're going away, which is
No,
a
absolutely.
testament to the partnership that's happening. So this is very exciting news as we're getting, John. You can go to a live feed and see our new building being built, the 70,000 square foot facility. I took a look at that yesterday, and now with Vitruvian under their leadership, John, the sky's the limit for us. It's very, very exciting. They're a $10 billion company. They are in the healthcare space. They have a wide portfolio. There's so much excitement in our ability to work with some of their groups in not only technology spaces, but even like marketing, and they have resources for HR, they have resources for accounting, all sorts of great tools and metrics and all sorts of systems and processes that could only make us better as we go forward in the future. So such a proud day, John. I know 8 years into this. I know you've been here at least a year with us, or a little over a year. And likewise, Mallory's over a year. But obviously, this is a moment that I couldn't be prouder of. I'm proud of the organization, proud of everybody that's part of the KCS team, and the sky's the limit. And this appears to be such a good fit for KCS.
Yeah, from my perspective, having been through purchases etc. before, this feels right. Where you got someone coming in who wants to invest, but let us operate the business as we plan to. But they're just there to help, and that really is what you want to hear from someone who is like I say, taking majority ownership.
Yeah, it's well that like you said, this is so exciting. We're finally making the news instead of having to talk about other investments. But I think there'll be more hitting the press. For those of you that are interested, you can find it all over the place. It's on the Business Wire. It's on Yahoo News. It's just everywhere, and it's so cool to be a part of it, John. Right?
Yeah, absolutely. Yep.
Yeah.
Yes.
And with that, I think that wraps up our news. We have a special guest today. She's been obviously active in part of our news. And so, as we sort of came to the concept of this podcast, as we move into our main portion, it was the idea kind of came from just, frequently we find ourselves having to describe or what we'll call introduction to bioanalysis and questions from a beginner, right? That's the name of our main portion. So with that, this is a good moment for Mallory perhaps to give us a little bit of her background. But before we do that, I think John wanted to give a little point as well.
Sure. Yeah. So this actually sprung up and actually it was Mallory's idea to do this topic, because we regularly meet to just discuss status of projects and prospects and things like that. And one thing that we as sort of very specialized scientists, we get asked what we do, and then it's a question of how do we communicate what we do to people who have no familiarity to the background of bioanalysis, etc. And we thought maybe it's a good topic to sort of talk about some of the basics and how we talk through what we do and how we describe it to other people. And like I say, this was Mallory was the one who planted the seed, and I then sort of passed it on to Dominic and said this could be a good topic. So here we are on podcast 39 doing this. So Mallory, do you want to go ahead and talk a bit more about your background and where you fit into everything?
Sure. Yeah. Thanks. Thanks for the introduction, John and Dominic. I'll start by saying I'm a loyal follower of the podcast. The episodes have really gotten me through these pandemic times we've had. So I'm very excited to be on and be a guest and be a part of the behind the scenes of it all. So a little bit about me. I've been with KCAS just over a year. And prior to that, I was really on the other side of the table, you could say, working in clinical research as a clinical research site coordinator at UNC Chapel Hill, go Tar Heels, and at Wake Forest University. So currently, I love it here at KCAS. I'm working in business development in the Mid Atlantic territory, living in Center City, Philadelphia. So maybe I'm looking forward to this episode with you guys. As John mentioned, this is something that I've been thinking about for a while. I've been, like I said, with the company a little over a year. I've learned the ins and outs of KCAS, all the many different things that we offer, and the second you put me, into a setting of being with friends and family or being home for the holidays, I found myself really stumbling over the classic question of, what do you do for work? What is bioanalytical? And
But
for
you,
me,
but
there's
you've
just,
also just quickly, you've also had similar discussions with uh, CEOs of companies where you've been talking to them and had that same question asked of
yeah.
them. So yeah, just
Yeah, yeah, I mean, exactly. Like I've encountered some higher ups like CEOs or COOs who these, it's you can say I'm in bioanalytical and biomarker support services. Okay, great. But really it's just what is bioanalytical? And I could go into a long rant, excitedly explaining what it is we do and what bioanalytical is, but for people who are outside this industry, it's just you kind of get lost. And so I'm just really curious, how do you guys handle that question of what is bioanalytical on a basic level?
Yeah, no, Mallory, I think this is something when we were preparing for the podcast, we talk about this all the time. There's, to me, it's an answer that gets a little bit hard. You say, even if people are in the field, sometimes that bioanalytical can be like a leftover, right? They're in the research area, they're really into research and they're doing maybe some development, but we're kind of picking it up from there. So that's where sometimes you get people in the CMC space or other spaces, they're just like, what is this bioanalytical? And then don't even get into biomarkers, right? That's a whole nother bailiwick. But like when I try to, you ask such a good question, like how do you describe it to like somebody who's more just say a common person? It gets different. I mean, sometimes I will. It can be as simple as, yeah, I work. Typically, what I'll say is, are you familiar with research and development? And people will say yes. Well, we don't really do any research. And what I mean by that is that's done by our sponsors. And that's where they're sort of evaluating what type of drugs or compounds or whatever sort of targets they want to go after or whatever sort of disease states. But at some point it starts to, as the drug gets into a certain part, it falls into the development. That's where we pick up things. And that's sort of where I lay the foundation of.
so,
And they say,
yeah.
well, what does that mean? And then I say, well, during the development part, at some point as the drug progresses, in order to get like FDA approval or regulatory approval, you have to have a third party do some of the testing for you. And now I'll stop there, but that's how I lay the foundation. And I think people that resonates well. It's like, okay, so and I talk about lead candidates, and we don't work on thousands of drugs at a time. We're really working on whatever is the one that's emerging as the one they want to invest in.
Yeah, I think my approach would be similar to, because people often come round and ask, and I'll talk, well, if you're taking any drugs, you know that those drugs have had to go through various trials to prove that they're safe and effective. And so, you'll have a trial with however many, you say, volunteers or patients. During the course of those trials, they will take blood samples from those people at set time points, but they need to know how much drug is in those samples. So they will send the samples to someone like us who will develop a method for analyzing that drug, and we will measure the amount of drug in those samples. Um, you know, we'll measure the amount of drug in those samples, and based on the concentration numbers that we pass back to the drug company, they can then model what the drug is doing. So in terms of how high a concentration is it reaching, how long is it hanging around in the body, and basically determine whether it's behaving how they want it to do. And then obviously there's other tests going on in parallel that are looking at the any side effects and things like that. So that's usually where I come from with that. After years of trying to hone the message to get it to a stage where people who really, it's trying to communicate on a level that people who have never encountered bioanalytical before might understand where we're coming from. And I don't mean to be patronizing on that. It's just that's where I'm coming from with like how to talk to them.
Yeah. Yeah, no,
And the
and
other,
Mallory, why don't you go ahead, Mallory, share some thoughts, and then there's some other,
yeah,
because it's not just the C levels. Sometimes there's small molecule people who have
Hmm.
inherited a large molecule and they're confused, and then
Oh, sure.
there's some converse. But Mallory, go ahead and share your thoughts.
yeah. I mean, I think, and that's a good point too. I'll definitely touch on that. I think the main thing I find as well is there's no, just with KCAS offering like the full spectrum of drug development, support, I guess you could say, is that like we do not have just one specific type of client that we go after. So sometimes whenever, I'm explaining this, someone will be like, "Oh, okay, so you just work with, pharmaceutical companies." That's not really the case. We support, and that's where it's, hard to convey. We support people who have, molecules or drugs or compounds that need bioanalytical work. There's no real black and white just because of how much we have to offer. Does that
Or,
is that
or
make sense to you guys?
Yeah, or delivery systems, things like that, So it might actually be, a company who have a more engineering approach because they're actually looking at ways of delivering drugs, but we're still measuring the actual drug itself. But they may be working on a drug that's already approved, but they're finding a better way to deliver that into the body. So yeah, you're right. It's not just your straight people developing innovative drugs. It could be innovative delivery systems and things like that.
Well, I think it even gets a step further as we broaden our way into some of our biomarker services and
Yeah.
Sure.
immunogenicity assessments, right? So John, we've been really focused on the PK part, and maybe we can touch a little bit about that in a little while. But there's so much after we, if someone is of interest, if I frequently I'll describe sort of PK, right? Like I'll just say, hey, and one of the things I want to emphasize is we're always looking at drugs that are put into species, either animals or humans.
Mhm.
And we take a sample before, and usually there's none. And then you sample the person or the individual every so minute, maybe it's 5 minutes, 30 minutes, hour, whatever. And there'll be drug levels that, and maybe there will be a lot. Then it slowly goes up, and then it forms this like PK curve. And I kind of describe and look at what a curve is, and then
Sure.
maybe 24-hour later it's gone. And in a biological setting, it might be a couple days, right? So there's just this, but it resonates with people. It's like, oh, that makes sense. And you talk about, but that's just the small piece of it. I think Mallory,
That's,
when
that's
you're
the
in a
easy
setting
piece.
trying to describe it to a CEO who's got a biologic, you then have to get into a different mode, right, Mallory? You have to start thinking about immunogenicity, and even regardless of the um, drug type, you always want to be thinking about biomarkers as well.
And like you said too, there are the cases where it's someone who's primarily large molecule focused, but something comes across their desk that's a small molecule. Or, there's also the reverse situation. So like those are such different beasts, but at the same time, I feel like there is somewhat of a common foundation between the two of just like, the building blocks of the PK and PD assays that it makes it a little bit easier to
Yeah,
translate, but that's still tough to go from large to small or small to large. And, I mean,
yeah.
do you guys have, like,
I
what's your
do.
experience with that?
I do. So if it's a small molecule person moving into the large molecule, I usually start to describe and say, hey, you're familiar with like a deuterated internal standard, and oh yeah, yeah. Well, that what we need is a capture antibody or an anti-id if it's a monoclonal antibody, right, Mallory? These are all terms you're getting
Mhm.
very familiar with, right? Or a positive control. So I kind of start to set the layer of, because the key piece of our industry, the key piece of bioanalytical is those building blocks, the
Mhm.
materials, the reagents we're using. We're only as good as those. So
Mhm.
small molecule people understand that. So I try to translate it large molecule wise. And then I go the other way. If I'm talking to a large molecule person who's now in an ADC space and maybe they got to figure out how to do a small molecule, I start saying, hey, you know how we really need to get a positive control? Well, you need this deuterated internal standard. And some might know what it is and others don't. And I harp on a deuterated. That's because my knowledge doesn't go any further than that with small molecules, right, John? But that's how, Mallory, I hope I answer your question. That's a good one. And that's how I sort of, because it resonates with them. They think, oh, I know what that means. I now
And
know. Yeah, go ahead.
sorry, and the other piece with small molecule is a key piece of the bioanalytical is the instrumentation. So it's less reagent based, like you talk about, but it's down to the chromatography and then the mass spectrometer, and then so again, talking to someone, you say we use this complex instrumentation, and it's really try and break it down as simple as possible. So chromatography is basically separating, the target analyte of interest from anything else that might be in that sample, and then introducing it to the mass spectrometer, which is basically a detector. So you have a way of you separate your analyte from the garbage, and you have a means of discriminate or of detecting it. I mean, some people will ask more, and then you go into what mass MSMSs. That it's if I can keep it off that, that's the better way to be. But
Yeah.
obviously,
Well,
we'll talk through that
I,
too.
I try to avoid Eliza MSD conversations at this stage because that's a platform thing. But you're right, John. Very good point. So Mallory, what are you thinking?
Yeah, I mean, no, that's all really helpful. I think kind of going back to my original point of, someone who might not be as familiar with this industry, another common question I get, which I'd love your feedback on, is, people will, whenever I kind of give the spiel, explain it as best I can, people will ask, oh, okay, so what drugs have you worked on? And, more recently, are you working on COVID drugs? And I think those questions, it kind of helps make our industry more relatable to those who are unfamiliar with it. So, do you guys get those types of questions? And are there like case studies you commonly share or anything like that?
If I can, I would
Sure.
actually ask people are there any medications you're taking, if it's a family member. I mean, I know I did this with my parents. My mum was a walking medicine cabinet because of all the conditions she had. But I also mentioned that I take atorvastatin, and we worked on atorvastatin in a number of studies. So I think I remember running, for atorvastatin, running a study with grapefruit, because that's one of the things they advise is if you're taking atorvastatin, don't eat grapefruit because it affects the metabolism of the drug.
Yep.
But that's a really good it's a really good example to talk to people about. This is the sort of thing that we're, we the trials, the analysis that we do supports that labeling on the actual drug bottles you have, and you like say avoid grapefruit because it affected how the drug was processed. So if that kind of thing is how I approach it, and on the COVID thing, I'd say yeah, we're working on a number of projects that are COVID related. Some of it's the actual therapeutics, some of it's the biomarkers associated with it.
Yes. So Mallory, I think John gave a very good overview, and he can get personal because he knows of one. But we've worked on over 300 approved drugs at KCS in our 40-year history. Many of them are in the small molecule category, and we do have some case studies I think we can dig up for you. I'm not as familiar with those being on the biologic side, but I know there's some pretty big approved drugs that
Hmm.
we did a bulk of the PK work on them. The other part is a lot of it's proprietary too, Mallory. You can't really reveal too much of it, so it gets a little
Hmm.
bit challenging with regard to that. But what we can share on the biologic side, we do a lot of co-med studies, so things like Humira and a lot of even like your PD-1 antibodies. We're doing a lot of studies with cetuximabs, all these really, I'd say of the top 10 monoclonal antibodies on the market, we have recently touched probably 8 of them. And that's in some form of a co-medication study. So somebody might, for example, have a novel drug, but they want to, dose with Humira, or, they want to give it to arthritic patients who are on Humira, whatever it might be. So then we have to subsequently study that as part of our PK study. So we actually have methods to detect those antibodies, but really we're focused on the innovator drug. So I hope that helps a little bit, Mallory. But we can really, and I don't know if that gave you a good answer, but I give you a list of monoclonal antibodies like nivolumab is a real famous PD-1 that we built a very good assay for. Um, and I'm not revealing anything proprietary because I'm not saying what client it is, other than, and we can't I can't share any of that data with anybody, but we do touch a large
Yeah.
volume of monoclonal antibodies. And then I'll end with the number, now the ADC space. It's such, we are working on many ADCs, and arguably we're, but again, we have to be careful, but we're working on one of the most promising ADCs in the planet right now. It's unbelievable. I think we're all aware of that. So that's a great story too, where we're actually bridging John's small molecule and the
Hmm.
biologic side on my side. So great stuff, Mallory. I hope that gave a good flavor for your answer in the future.
I think
Yeah,
the confidentiality thing is definitely part of that, because we're all, after years in the industry, you're always really cagey. We're always really cautious about saying what we've worked on. So, I mean, I will talk about drugs that have been on the market for years, and so it's really not nothing. There's nothing too,
Yeah,
we're not
correct.
really giving too much
When you
away.
meant,
It's
yeah,
when
and we can dig up Mallory, there's some that our names are on those submissions, so that's public information, and we could probably
Hmm.
dig up that for you.
Or we've
Yeah.
published papers or posters or things like that. That's another thing, because then it means it's been out in public that's saying we're associated with something like that. So
Yeah,
that's
and
that's the way I would tend to use as a measure. I,
it ties our company back to make it relatable to people,
sure. Yep.
people outside. But I mean, you did bring up a good point earlier too, John. I've spoken about with you before. There have been, some high level people who, like CEOs or COOs, been in a conversation with them. They're obviously very much in the industry, but they admittedly don't really understand what bioanalytical encompasses. And so, once we get down a little bit more into KCAS service lines and how they can help support their programs, you're trying to explain it from a level of bioanalytical newbie, I guess you could say. And so, I mean, I'd be curious to hear, how do you guys explain, like, just general PK, what our PK is, and even, our discovery service lines.
John, do you want to jump in that,
So,
or?
I mean, discovery, I would tend to say this is, the very early stage where you're trying to identify a candidate. And again, I'm coming out from a small molecule perspective. You may have a number of, you've identified the target. You may have a number of, a, a number of molecules that you think may have potential. So these are your early PK tests in animals, with no regulatory sort of need to them. You're just really trying to identify the one of the group that might have function the best for to then identify going forward, going into more GLP, and that becomes your lead candidate going forward. So it's really that early stuff. And I mean, a lot of discovery is just determining solubility, the sort of early interaction type type screens. So I
Yeah.
mean, discovery is quite a wide piece of it, but again,
Well, I
where
think
we are, we're very focused on the PK
Yeah,
piece.
John, I think you nailed it. It's like somebody will have basically, it could be thousands of candidates, right? Because you're talking
Yep,
about just, and
absolutely.
it could be as simple as a bending of a carbon here, and that, and then suddenly you got a different product, right? So then even in this ties into the biologic space. Um, we call them I don't really love the term, but I'm going to say it anyways. It's a quick and dirty assay. So really, what you're, like you said, you're looking for maybe some solubility, and we're just trying to get a nice reproducible range that we can detect. And we also typically, it's kind of like, it's the initial phase to lead into a future true PK study. And what I mean by that is you sort of start to look at the Cmax in these early stages, so you can get a sense of, because basically whatever say you have 100 of these products, eventually you're going to make it the same way. So it's going to have some similarity across all 100 of them, so that when one does emerge, you get a lot of information from that quick and dirty, we call them research grade assays that we might make. And on the biologic side, we do have some discovery services using what we call a generic or a universal assay. And we can screen lots of monoclonal antibodies because, without getting into too much detail, we could screen 6 or 7 of those, and really with one method that can be used over and over again. So, Mallory, I hope we're touching a little bit, or at least helping you understand some of that research stuff in terms of how we try to position it. But it's a question that requires, in my mind, some back and forth with the individual asking it to allow me to properly answer it. Right?
Yeah.
I kind of got to probe this person to see how much they know to help break it down for them.
Yeah. And I think even, just a recent conversation I had that touches on this, again, unfamiliar with the bioanalytical, he actually asked just flat out, why would I need biomarkers? Like, why would I need your PD or biomarker services? What would that do for me? What would that do for this drug? That just a flat out question like that. And I know, Dominic,
Yeah.
you're all over that.
I'm already like my head exploded, right? Like,
I've just added another half hour to the podcast. Well,
Yeah.
Dominic, talk through
like
that.
boom. I mean, for me, if someone were to say that, I think if you have a biomarker, your success rate increases. I think it's 35%. So it has a bigger success rate. I think of all the drugs approved last year, I haven't seen some. COVID kind of mixed up our stats a little bit, John. We used to report on stuff like this, but at one point it was like, 5 years ago, you go back and look, and the number of approved drugs that had like a PD biomarker was like less than 10%. Well, flash forward today, it's well into the 30s and 40s and only rising. So what it really helps you do is a couple things. One, it helps. I call it the biology or mechanism of action. It helps support that. So if your
Mm-hmm.
drug's targeting something and you're supposed to bring down a given something, some sort of inflammatory marker, or you're supposed to raise up something that's protective, there's a measurement for you, right? So some
Yes.
sort of fluctuation. And by the way, it could be as simple as your heart rate. That's a biomarker, right?
Yep.
But in some sort of soluble, typically it's maybe something that's produced or secreted, or it's something that's present in not normal. Most of us don't have it. Cancer patients will have high levels of it. You give the therapeutic, it drops. So that really helps with functional and some sort of mechanism of action. The other piece is it helps you stratify your data. So say you've tested 1,000 donor individuals or patients, and maybe like, hey, only half of them are responding. Well, if of those half, 80% of them had the biomarker, now you know why. So it helps stratify the data. And so maybe you can use it as, maybe I don't treat everybody with metastatic renal cell carcinoma, but maybe I treat a population that has this biomarker. So what that means is if a drug can't be all inclusive, at least maybe it doesn't fail, maybe it can treat a subset of patients. And so that's how I kind of look at the value of biomarkers. First and foremost, it's for the patient. Let's be very clear about that.
Mhm.
I
I
don't
mean,
know
at the
anything.
most
I
basic
totally agree
level,
with that.
you're saying this, you're basically whatever it is, you're trying to prove this is what the drug does to the body.
Yeah, the
Is that
mechanism of action, right? Yeah,
Yeah,
so exactly. How is it augmenting the patient? So the PK, that just shows, oh, it wasn't there. We gave it. It goes through their body. They release it. They secrete it. They absorb it, blah, blah, blah. But the biomarker component shows that it has it can't, we can't get in and see, oh, hey, these cells are doing this. But it has the ability to say, hey, we are augmenting this individual's immune system if we're causing a pro-inflammatory biomarker to get knocked down, or if we're, because that's stopping whatever cell was producing it. Similarly, if it's a protective thing, it's the other way around. Oh, these cells are making it. So maybe
Mhm.
is that I hope I'm giving it to you the right way, Mallory, right?
Yeah,
Is that
that
okay?
definitely helps. I wish you had been there the other day. This
Hey,
right.
that's why you're here, Mallory. That's part of it, right? I mean, you know how to answer it, but you really were hoping to hear it. You want to hear it from, hey, I've been studying these for 25, 30 years now. Don't beat yourself up, Mallory.
Yeah, I mean, it's interesting because, there is some level whenever you are within the company that's developing the drug, they obviously have a lot of knowledge on it. But I mean, I've just come to realize like this specific little bioanalytical piece, it's a very specific area. And so I think that's why, KCAS is so helpful because we specialize in that one little area. There's a lot to developing a drug or, compound, anything it's specialized. And so I think that companies like us, that's what's so good about it. They don't have to bring those people into their own company. They can, rely on us.
Yeah.
So that's, I
To
mean,
your
I
point,
think
Mallory, the inception of a small biotech, or they're going to spend their first 4 or 5 years doing all research, and then they're going to spend a couple more years getting into that CMC space. So you might be 5, 7 years into a company, you haven't even thought about bio. Maybe it's shorter, a year or two, whatever the time frame. But you could be a couple years into a journey, right? And then suddenly someone says, hey, you got to look at bioanalytical. They're like, what's that? Well,
yeah.
that's when you start putting it into stuff and looking at it in matrix. So they've been looking at it all along from a efficacy standpoint. John talked about solubility. They've been looking at it a wide variety of ways, and then the manufacturing process is so focused on buffers or solutions or diluents or whatever you're going to store it in or whatever you're going to solubilize it in, that the intensity's there. And frequently you're talking to people who are, they get to a certain point and maybe they've handed it off to another team. And so maybe I've been working in the drug development, the research area for 30 years, but for 25 of them, I just call my colleague Susie and she takes care of the bioanalytical. And that's who you're encountering. Suddenly they got a responsibility and now they need to learn about it. I think that's what happens.
exactly. So, I mean, looking at you again, Dominic, probably the most complex piece of, KCAS and what we offer that's complex to outsiders and even insiders. How do you explain the need for our immunogenicity, ADA, NAB types of support services to someone who's, again, somewhat unfamiliar with the specifics of it.
Or even yet, I'll say we frequently hear people say, "Well, I don't need that because my drug doesn't cause an immune
Yeah.
response." But somewhere
Yes.
along the line, the FDA asked them to do it, right? So you're the not, and you're right. The concept of immunogenicity is it's a word that was coined maybe under 20 years ago now. Sadly, they used to be called HAMA responses, human anti-mouse antibodies. So when we're starting to give antibodies to people, we just characterize them as human anti-mouse antibodies, human anti-chimeric. We had HACAs, and then therapeutic, human anti-therapeutics, HADAs. So all of that now is the term immunogenicity. I gave a little history lesson to you, but that explaining it to a novice takes a little bit of patience and then a little bit again probing into like how familiar are you with small molecules in general. You don't have to worry about it, although as you start to tag a small molecule onto a biologic, there's some pause there. We call it a risk assessment. So that's the word. You've heard me say that to somebody. I'll say, hey, have you done a risk assessment of your drug? And if they say no, that's when I'm kind of like, well, you really probably should either get a consultant or somebody to help you do that. But then the need comes from, because I think that's your question. Why do we need it? Is that your question? Why do we need immunogenicity, Mallory? Or what?
Yes.
Sorry.
Yes.
Yeah.
Yeah.
So the reality is when you put a large molecule, and let's say anything over 10 kilodalton, you get into the space where the immune system can start to recognize it. Anything smaller than that usually just passively gets taken up and can be excreted through the body. These larger proteins or peptides, there's a couple things that can happen. They can, if there's any sort of clump, they agglutinate or clump, that can trigger an immune response. Sometimes there's a couple things, right? You can immediately put it into somebody and immediately the innate immune system, and that's some sort of like, adverse event or an anaphylaxis or something of that nature. If it's that gets way out into an extreme. But that's the first challenge with a protein when it goes into the body is, does it trigger, I think of it as the innate immune. And then over time, if the body starts to say, if by some degree it starts to clump or it starts to form a complex within you, then that's going to maybe trigger an adaptive immune response. And that's what we're really talking about. An immunogenicity response that's specifically so immunogenicity is a big word, but if we drill down just into the anti-drug antibody, right? ADA methods are what we do a lot of. That is when, again, the body makes an antibody against a drug, an anti-drug antibody. So that has to be mediated through an adaptive immune response, has to have some sort of recognition by an antigen presenting cell, then subsequently some trigger of an adaptive immune response. So some sort of T helper responses, or maybe even somewhere in there, you're going to get B cell, without going into an immunology lecture on you. But the adaptive immune response, the output is a plasma B cell, or B cell that will be producing antibodies against the drug. A lot of times this is completely okay. It might be something that happens because it's sometimes the if you give a protein, it might be seen as foreign or not non-self, so the body might make a response, and then a few weeks later it's gone, and then you have no problem, and maybe it's never even resolved. So, but other times it can have effects where you make an antibody against it, and then it can neutralize it, and that becomes a problem because then it stops the efficacy of the drug. So I'm take a small breath. I hope I explained some of that to you, Mallory, but it's a big question because I haven't even gone into the nuances of the tiers of the ADA assay, which you know a lot about, right? The screen, the confirm, the titer, and that might be where you're getting. Like, how do I get from this individual not even knowing what immunogenicity and an anti-drug antibody is to the realization they need to invest heavily into a
Yes.
positive control, right? They need to make a polyclonal sera and purify it. So they got a positive control, a PC, and then the realization that they actually have to do a screening, confirming, and titering method. And some of them will come out and say, oh, I need a NAB assay. And you're like, boy, you need way more than that, right, Mallory?
Yes. Yes. That I mean that's really helpful. These are the types of questions that I think people who might even be in the field have, who, you don't want to just say, nope, I'm very unfamiliar with bioanalytical. So I think this is relatable. All of this has been really helpful for me. I'm in the field, and I have friends and family who ask about it. So it's helpful for me. It's helpful for those people who are outside the field who don't understand what we do. And then, like I said, the high ups in the companies who have these promising drugs, but they don't know where to start with the bioanalytical and biomarker work. So I think this is a necessary conversation for anyone just wanting to dip their toes in the water, or really understand they have this drug. What do we need to do with it? And then for me in my conversations with people. So yeah, this has been really helpful. I really appreciate you, John and Dominic, really taking the time to talk through this. And I'm sure that you've experienced this, in your own personal lives or in your professional career with people. So I'm really glad that we were able to, kind of talk through it.
No problem, Mallory, but you're not out of the hot seat yet. I'm going to give John any last thoughts, John, before we
No,
start.
I think it's there's obviously the pieces of this that we're both comfortable with. Dominic on the large molecule, me with the small, but this industry is evolving at such a rapid pace. We get confronted with pieces that we're totally unfamiliar with, and we need these explanations too. So the ADA piece, it's great to hear Dominic talk through that, because that helps formulate it in my head. It's
the funny part about ADAs is we didn't even like, most of the time I get asked what are the regulations, and that makes my head spin. So we're still scratching the surface, because that you want to talk about something that's constantly evolving. It's the regulations around, this is still somewhat in flux in terms of how much you really need it. I'll throw a curveball at you. You could technically eliminate all ADA testing by just looking at your PK method, and unless there's some sort of PK profile perturbation, you could assume you don't have any immunogenicity, and that's what the field hopes to move towards. Unfortunately, that is not going to happen, because there's too much risk. If it's a non-neutralizing and you're producing a copious amount of antibody, you want to know that, even if it's not neutralizing, right? So
Sure.
I hope that made sense, especially as you dose with more and more. I hope to be 110 years old and I'm taking 5 different monoclonal antibodies, right? So we better know if you've got these types of drugs against you, because that gets into a whole nother speculative sort of answer around what would be the impact if I had an antibody against, Humira circulating against cetuximab. That could be potentially quite risky, right? So that's why I hope it made sense there what I said, because you could just look at a PK profile and say, oh yeah, there's no immunogenicity. But anyhow, obviously a lot of thoughts for me here, a lot of passion around this. But Mallory, I think before we get you into the hot seat, we're going to talk about next week's episode, which is, John, podcast number 40 for us.
Exactly.
With that said, John, what do we got? We got a big surprise next week, right? But we're just going to tease you with that. So for podcast 40, we're going to surprise you with something. Isn't that right?
It's going to be a very different situation. That's all we want to say. And
so then we're going to turn our focus to Mallory. Hot seat questions. This is a pressure cooker right now. What's your favorite podcast, Mallory? Are you ready for it?
Dominic lives for this stuff.
There's a right answer here, and I think it's the Weekly Bioanalysis, of course. But I think if I had to pick another one,
no, we're talking about our episodes, so not podcasts in general. What is your favorite weekly? Maybe you don't know one, right? Maybe you think about one you like. Yeah, you don't have to give us the number, but what episode if you can't recall the number? So I was like, we're looking for. Did you like podcast 10, for instance?
No, I don't remember the number, but it was the one with my favorite colleagues, other than you guys, Jeremy and Chris. I really enjoyed that, and that kind of felt like it opened up the field to bringing me onto the podcast. I really enjoyed that, because they're my colleagues. I work alongside them, so it was very relatable for me. But yeah, I like the end of the episodes where Dominic always talks about what he's going to make on the weekend, and that's pretty much every episode. So
actually,
the pizza oven, I'm really looking forward to seeing some pizzas you make out of there. That's interesting to me.
Well, maybe it still hasn't warmed up enough here. I haven't been able to break it out quite yet. You got to be over 70 to, they say, outside to use it, or you're not going to cook it the right way. And the last thing I need to do is use my pizza oven and fail, right? But
you should just space generator, heat generator.
Yeah. By the way, we're not going to let you off the hook with that.
No.
Canned answer around who you like the one with your colleagues on it. That was, come on, I'm just kidding, Mallory.
So yeah, the big question for us is who's going to use their oven first? Will it be Dominic using his pizza oven, or will it be us using our replacement oven, which is still in our basement after, actually we're getting on for 6 months without installation.
Oh my God.
That is so you can't get anyone out there to install it, John?
We got the installers. We can't get the inspector. Last we heard, he disappeared down to Florida, and he'd be back sometime in April, May, or June.
Oh,
So
good Lord. I got to call my cousin Vinny. Just put it in. Don't worry about the inspector. No, that was a joke. I don't have a cousin.
no, I realised that. So as we're talking food, Mallory, what's your favourite dish to make?
Well, lately I have really enjoyed making roasted Brussels sprouts. I know that sounds gross, but with sesame oil, honey, sriracha, and soy sauce, and sprinkle sesame seeds on top. That is a wonderful dish.
Oh man, I don't think that sounds gross at all.
No.
It sounds great. I'm a great believer in the combination of Brussels sprouts and like baking them or pancetta.
See,
Some onion in there. That's like,
I love veggies, but I like to put them in the oven and cook them until they're almost a crisp. So I don't really know how nutritional that is for you, but it's phenomenal. The same with kale chips, asparagus, things like that.
Well, with the Brussels sprouts, you're going to get the ones that are like chip-like on the side, and those aren't very good. But the heart of it, I usually cut them in thirds, depending upon the sides or having them. And then all the stuff that falls off is delicious. Yes, that's not good for you, but the bulk of it still is very healthy, I would think. And Mallory, you struck a chord. I have a bag of Brussels sprouts. I usually make it once a week. I toss it in olive oil. I use sriracha, interestingly enough. And then I use a rub that I like. Whatever type of rub I have, I'll sprinkle it with like a barbecue rub and toss that and then bake it. So similar, interesting.
But
I think it's fascinating. I should have guessed it that you would go there. But I think it's great. And I might be interested in your Brussels sprout recipe to try it. We're going to move on from, go ahead.
No, it really does.
What's that?
Cooking Brussels sprouts. The smell is awful.
You can light a candle for that. My wife she got mad at me last night. I steamed broccoli. I was like, don't worry.
See, the one smell I don't like while cooking is salmon. I really cannot stand the smell of salmon cooking.
No, I'm very picky. So
the smell of salmon just sticks around for weeks.
Yeah.
I'm a classical eat anything. The smell doesn't bother me. I don't care.
I didn't say I wouldn't eat it. I'll eat anything as well. I love fish as a meal. If I go to a restaurant, I'll routinely go for fish rather than anything else. But salmon cooking is it's something.
I was gonna say, if I want salmon, I'm going to go out for it.
Yeah,
you guys are all wusses. It's like you sound like my wife. Just so funny. By the way, Aaron has asked for your recipe, Mallory. So I think you should maybe post it when you get a chance. But you're not off the hot seat yet. What is we'll take a little more time. We're running long, but this has been a lot of fun having you on as a guest. What is your favorite adult beverage?
That's an easy one. I really like a good tequila pineapple. And I can give some background here because pineapple has anti-inflammatory effects and it's healthy for you. So it counteracts the alcohol and you feel fine.
Okay. If you drink 6 of them, does it still counter it?
Yeah, that's the problem with red wine, isn't it? Red wine's supposed to be benefits, but you never stop drinking it at just one glass.
No, but that is true. I'm just very curious of if it actually makes a difference. But yes, pineapple has anti-inflammatory effects and it counteracts the alcohol. So that's in my head, that is just the perfect go-to drink.
All right. Well, you'd like a good Pearl Harbor then. Mallory, you gave a shout out earlier to your Tar Heels. I'm not going to let you off the hook without mentioning that Mallory was a Division I soccer player. At least you were. You'll be humble about it and say, "Well, I was only on the practice squad." But as somebody who loves to compete athletically, I never played or made any field Division I. So I think I want to give you a big shout out there. So that's why you're still very health conscious about it. But
practice. Yes, but thank you. I appreciate it.
Practice or otherwise, most of us could only dream of being in any sort of Division I setting. So I think it's important to mention that about you.
But they have news as well. with the new coach and whatnot. That's pretty exciting news. So for basketball, I hope you all know about this.
Oh yeah,
Yes,
Okay.
right.
Okay.
Who replaced Williams? Who was it?
it's Hubert Davis.
Oh
Yep.
yeah, longtime
He's been
NBA
here
player. I like him. Mhm.
from Chapel Hill, so it's a good fit. But no one can replace Roy. Let's just be honest.
Well, he still
Oh,
broke the hearts of Kansas when he left,
yes.
although we inherited Swift, so we're okay. But yeah. How about
See,
some weekend plans? Oh,
I'm
go ahead.
much more Dean Smith era Chapel Hill. So
Yeah,
Yes. Yes.
likewise.
That's fine.
So, last question for you: what are your weekend plans?
Well, I just had a birthday, so I will be celebrating my birthday with some friends. I'm actually really excited about that. That one of them works in healthcare, so I really have maybe seen her one time in the last year. But she is fully vaccinated now. I am as well, so we're going to maybe get a drink or something together. But for me, it's not a birthday; it's a birth month. So I have the month to celebrate. So this will be weekend 2 of birthday celebration. So I'm very excited about that.
Oh, that's great. We do have birthday weeks here. At least my wife likes to do a birthday week. I hadn't heard of a birthday month. That's
It's
great
birth
news.
month.
Yeah.
Yes.
And so,
I, I,
are you able to
I'm
go out in inner city Philly and everything? Is that where you guys are hanging out?
Yes, outside has pretty much been open the whole time. They've really made like the outside of restaurants and bars just as nice as the insides.
Mm.
So and with the weather being nice, it's a good treat. But I
When you
will
say it's
say
nice,
indoor dining has opened in Philly.
sorry,
So
repeat
that's
that. Go ahead and
um,
repeat that.
indoor dining has opened in Philly,
Oh,
which living there for a year, I've only been in a restaurant 2 times. So that's big. I'm
when did restaurants open up?
Yeah, yeah.
When, no,
There's
when did they open up? Like last week or?
no, I wanna say they opened at reduced capacity about a month ago,
Wow.
almost. Yeah, but I
Really?
mean, when I say reduced capacity, it was like 25%. So downtown Philly, it's slammed, you can't get in places unless you make a reservation 2 days in advance, I'd say. It's
Oh
it's really
man.
something.
I
so
mean,
so
Jeremy and I are shaking our head in laughter because we've been open at that capacity. We've been open since like, I don't know, July of last year. So we're living in different worlds.
Yeah,
so
Yes.
so Mallory, if you can get a reservation, Zahav is really good. The Israeli restaurant, which is down in Old Town.
Really? I think I
Yeah,
have heard of that.
yeah, and we also like the Han Dynasty, which is
Yes.
Szechuan, and again the one in Old Town is really nice.
You're too
So
sophisticated, John. Just go to Geno's and get a cheesesteak, huh?
Oh,
obviously my son spent 5 years in Philadelphia, so
that's
we know a
great.
number of the restaurants, etc. I think I've pretty much stayed at every hotel in the city. So yeah, there's some good places in Philly.
Well, now that they start opening, I'll bother you for
Yeah,
recommendations.
I'd love to
Last
get back
year.
to I'd love to get back to Philadelphia again. I really do enjoy my visits there.
Well,
Love
hopefully
Philly.
before 2021's over, we can get out and see some people in person and you
Yeah.
can come to Philly.
That's one thing we didn't touch on, in terms of your background. I mean, you said you've been with the company a year, but effectively your job it hasn't been a typical business development job because it totally coincided with COVID. So you've never
Yeah.
actually met a client face to face in that
I
in that
feel like
time.
the guinea pig. Yeah. I mean, it's been all virtual. My job has been virtual for a year. So it's really interesting. It's changed the way we do things, that's for sure.
John, we're running long, so we're gonna skip what you and I are doing. I'm have nothing really going on or exciting, so I won't share anything. It's supposed to rain here all weekend, so I'll have probably a lot of videos or maybe even get caught up on some work. John, what about yourself?
Similar deal. It's going to
Yeah.
be a quiet weekend here, so I think it's yeah, just take it easy, and yeah, hopefully the temperatures aren't going to be great here.
Yeah.
Like say snow today, although it seems to have all melted. But yeah, quiet
Well,
weekend.
Mallory, congratulations for being out of the hot seat. You're
Oh,
officially off the hot seat, and I'll just my parting thoughts are: it was great to have you on the show. I think you've been a wonderful addition to our BD team. You've done an amazing job done there as a BD representative for us in a very let's call it tumultuous year. And even I'll give you a big shout out. I think you exceeded your goal in Q4, knocked it out of the park for us. So you're on your way. It's been such a joy to work hand in hand with you, and look forward to more. And John, any parting words from Mallory as we wrap
No,
up podcast number 39?
I totally endorse everything that Dominic said, and I look forward to getting to the stage where we can visit customers, because like say Philadelphia is an easy drive for me, and I'd love to get back down there, so we'll actually get out and promote KCAS face to face.
Well, thank you guys. I'm honored to be on this podcast with you. And, this last year, I truly could not have done it without both of you. So I apologize for my daily phone calls and needing your advice on things. But, that's kind of my job, is to bother you and get your help as much as I can. And you guys have been great. So thank you.
And it's no bother, and you're not alone. You don't look at it that way. We got BD reps that have been with us for decades, and they still call daily as well. So no need to apologize. It's all good. It's in the spirit of what I like to call winning, right? But Mallory, thanks, and that's a wrap for podcast number 39.
Looking forward to the big one in number 40.